Dear colleagues, first of all, I would like to say that it's a great honor for me to participate in such an event and to present the latest results of our study. Our study is devoted to targeted genome editing on small nuclear RNAs in human cells. The study has been conducted at the Laboratory of Genome Editing of the Institute of Chemical Biology and Fundamental Medicine in collaboration with the Novosibirsk State University. Small nuclear boxy RNAs are a subclass of SNO-RNAs, which are involved in post-transcriptional maturation of ribosomal RNAs in human cells. They guide site-specific ribose methylation of nucleotides in ribosomal RNA. They contain conserved elements in the structure, boxes C and D, and in several cases they also contain an additional pair of boxes C and D. These boxes are very crucial since they are conserved elements, and they are crucial for recruitment of SNRNA-associated proteins, which altogether form so-called small nuclear ribonucleoprotein complex, or SNO-RNP, which is a functional complex. There are also guide elements between the boxes C and D, which is complementary to the region within the target rRNA molecule. And if you take a look at the left side of the picture, you can see that this is a fragment of human 20A subunit ribosomal RNA, and it contains a lot of modified nucleotides. Most of the modified nucleotides are presented with ribose methylated nucleotides. Guide sequence is usually 9 to 21 nucleotides long, and the nucleotide to be modified in the target rRNA is complementary to the 5th position to the box D sequence. As adenosine methionine serves as the donor of the methyl group, and as a result, the nucleotide is methylated at the 2nd position of the ribose moiety. Well, I suggest that CRISPR-Cas knockout mechanism requires no repetitive presentation. What's important in our study is that since the majority of SNRNAs are encoded within the introns of other genes, the use of CRISPR-Cas9 systems allows one altering the genome sequence of a SNRNA of interest while preserving the exon structure of its host gene. And since SNRNAs have a lot of different structural elements in the structure, and depending on the presence and location of the palm sequence in the SNRNA sequence, double-strand DNA breaks can be targeted at various elements of SNRNA. And the obtained cell lines can be further used for the functional study of SNRNAs in human cells. The aim of the study was to develop the strategy and the systems for genome editing targeted at specific regulation of the activity of SNRNAs. And the main purpose of the study was to obtain and analyze SNRNA deficient cell lines. 3 SNRNAs, namely U74, 75, and 77, they were chosen as the targets in our experiments. These SNRNAs are encoded within the introns of the GAS5 gene, alongside with a series of other SNRNAs. And double-strand DNA breaks were targeted at conserved elements boxes C and D, denoted with an asterisk. The experiment itself included the following steps. First, single guide RNA design, then construction of the expression vector, transfection of human embryonic kidney cells with the obtained vector, then isolation of clonal cells, analysis of the obtained clones using various methods, and then we performed analysis of the expression level of target SNRNA, analysis of the level of target nucleotide nucleotide modification in ribosomal RNA, and differential analysis of differential gene expression by RNA-seq. Forgot to mention that considerable amount of our study was also performed by Anastasia Matveeva, and if any technical questions considering the technical aspects of our experiment arise, they can be addressed directly to Anastasia today during poster session. Her Boston number is 23. We managed to obtain clones for each of the 3 target SNRNAs, and as for the monoclone for U74 SNRNA, these cells were characterized by low growth and survival rate, and as for the 2 other SNRNAs, we managed to obtain monoclones for them, with each carrying the mutations, and the obtained monoclones were analyzed for the nature of the mutations using TIDE software and TA cloning followed by Sanger sequencing. Analysis of the expression level of target SNRNAs shows significant downregulation of target SNRNA for each of the 2 clones, clone 75-2. We can see that U75 SNRNA is significantly downregulated, U77 SNRNA is significantly downregulated in the clone 77-1. There is also a slight downregulation of U77 SNRNA in the clone 75-2, but neither of these mutations affected the level of U74 SNRNA, which is located upstream in GAS5 gene in relation to the other 2 SNRNAs. This might be due to the fact that U74 SNRNA and the 2 remaining SNRNAs they undergo different assembly pathways, while U74 SNRNA undergoes splicing independent assembly. U75 and U77 SNRNA, for them, splicing dependent mode of assembly is typical. Next, the monoclone with 2 deletions have been selected, with 2 deletions in U75 gene has been selected. It contained 2 deletions. One allele contained deletion of 9 nucleotides, while another one contained a deletion of 10 nucleotides. Well, the question arises, does one nucleotide can make a difference? Because we performed RNA-seq analysis, and it demonstrated us that only one mature mutant form, this one, is present among mature forms of this RNA, while the other one, this one, is absent. RNA-seq analysis also demonstrated us that the total level of U75 RNA was decreased 2.3-fold compared to the control. We suggest that this might be due to the fact that deletion of an additional nucleotide, that's making it 10 nucleotides, makes the guide sequence too short for this RNA to be properly matured and processed into mature form and properly functioning. Next, we also isolated the monoclone with mutations in U77 RNA. This clone carried 2 mutations, an insertion of 1 nucleotide denoted in red, and deletion of 11 nucleotides. RNA-seq analysis showed that both forms are present in the mature transcripts in these cells, and with the first one, this one being more abundant than the other one, with a ratio of approximately 50-1. RNA-seq analysis also showed us that the total level of U77 RNA was increased significantly, approximately 8.5 and 8.5-fold compared to the control. So the same question arises again: does one nucleotide can make a difference? If we take a closer look, we can see that this insertion is located at the C-box adjacent area, and this is the terminal area, the terminal region. And this terminal region, it interacts with the other terminal region of this RNA during maturation, and it interacts it's complementary, and it results in the so-called K-turn motif, which is very crucial for recruitment of snRNA-specific proteins or nucleolar proteins. And what we see here is that these adenosine, denoted in red A, addition of this adenosine, it is beneficial for this snRNA, and it doesn't change its target specificity, since the guide sequence is located in here, and we have an insertion of nucleotide in here. But it definitely should improve, should be beneficial for this snRNA for its stabilization and further procession, assembly into snRNP complexes. Next, what we did, we assessed the level of target nucleotide modification. Since snRNA have the targets, so we need to find out whether there were some changes. We used the method of reverse transcription followed by polymerase chain reaction, and this method is based on the ability of methylated nucleotides, methylated by ribose, to terminate reverse transcriptase with the formation of truncated cDNA products with a length corresponding to the position of the modified nucleotide. This situation takes place in case of using suboptimal or low DNTP concentration, and the outcome, the yield of these truncated DNA products is the highest when using suboptimal DNTP concentration, while in case of using optimal DNTP concentration, the yield of such products is the lowest, and we have longer cDNA products. Next, if we use modification-specific primers, and we perform real-time PCR, in case of truncated cDNA product, it won't be amplified, and longer cDNA product will be amplified. So in that case, we will have amplification products. And what's important for this experiment, in this method, is that the level of methylation is inversely correlates with the level of amplification. If we take a look at our clones, what we need to do is to perform the reaction at two different concentrations, optimal and suboptimal, and then to compare this reaction for our clone and control cells. What we see here, we see that we see an increased level of amplification product for our clone, which is indicative of a decreased level of methylation of our target nucleotide, in case of clone 75-2. And as for the clone 77-1, no changes are observed. Analysis of differential gene expression in the obtained clones showed significant upregulation on the number of genes in the poly A fraction in the obtained clones. Functional analysis of RNA sequencing data shows significant upregulation of a group of genes involved in innate immune response and antiviral immune response. And in case of the clone 77-1, we can see that the group of such genes is smaller in the total pool of upregulated genes, and we also had a group of 25 genes which were commonly upregulated in both of the clones, and most of the genes were involved in innate immune response and antiviral response. Upregulation of the innate immune and antiviral response genes in the obtained clones, as we suggest, can be due to the inability of the mutant snRNA to be associated with nucleolar proteins, and in case of canonical snRNAs, they are associated with nucleolar proteins, and then they stay within the nucleolus where they perform their function. In case of mutant and bound snRNAs, they are released into the cytoplasm, where they cause the creation of the innate immune and antiviral response through the protein kinase R mediated pathway. Thus we obtained snRNA deficient cell lines, stably expressing snRNAs with altered structure. What we observed is that, first of all, U74 snRNA, as we could see, is crucial for cell viability and survival. That cells like in native form of one of the two snRNAs, namely U75 and 77, are viable and metabolically active, Box D is the most efficient target for generation of snRNA deficient cells. Crispr-Cas9 mediated knockout of snRNA results in decreased level of methylation of the target nucleotides in cells, and CRISPR-Cas9 mediated mutation in U75 and generation of mutants U75 form in the cells results in upregulation of the innate immune response and antiviral response genes. I would like to express gratitude to our colleagues, laboratory autonomy team, head of the laboratory Valentin Viktorovich Vlasov, head of the project Grigory Stepanov, next to our colleagues from the laboratory of biotechnology, supervisors Vladimir Richter, and especially Dmitry Simonov for performing bioinformatic analysis of RNA sequencing data. Our colleagues from the Institute of Cytology and Genetics and the Institute of Molecular and Cellular Biology, Novosibirsk, for the help with cell experiments, especially cell sorting, and also a group of researchers, a team from Kazan University for RNA sequencing analysis, as well as RFBR grant for financial support. Thank you for your attention and your
Спасибо большое. Если есть вопросы, пожалуйста, задавайте. Мария Андреевна.
Вы не могли бы сказать, а какие гены, хотя бы примеры из innate immunity у вас отрегулировались? Ну, да, ТЛР, ВИК, интерферон.
Интерферон альфа, интерферон бета.
Понятно. И в принципе, планируете ли вы сделать какие-то эксперименты, которые позволят физиологически на ваших клетках увидеть изменения в иммунном ответе? Ну, я не знаю, заразить вирусом, еще что-нибудь такое сделать.
Для чего?
Ну, для того, чтобы, ну, на самом деле изменение экспрессии генов, да, это, конечно, здорово, и это вам некое направление, в котором надо, наверное, думать, да, но без функционального теста обычно, ну, работа с функциональными тестами, она обычно красивее получается, собственно говоря, поэтому.
В следующем этапе нам нужно верифицировать гены, которые...
Ну, это безусловно, да. Понятно, спасибо.
Есть ли еще вопросы?
Да.
Пожалуйста.
Да, у меня короткий вопрос вот, к самому подходу. Значит, известно, что некодирующая РНК инактивировать нетривиальные задачи, сложнее инактивировать, чем ОРФ и кодировку белка. Вот вы использовали вот этот NHEJ подход, да, когда у вас там в одном аллеле что-то одно делетировалось, потом в другом другое, и вы там смотрели. А вы не задумывались о том, что, может быть, лучше было бы все-таки использовать гомологичную рекомбинацию, сделать прицельную такую модификацию, которая бы разрушила вторичную структуру ваших некодирующих РНК, и это было бы более правильной моделью для изучения вот, как бы сказать, нокаута вот этих РНК?
Да, спасибо, это очень хороший вопрос. Я хотела просто рассказать, что тут это первые эксперименты, которые мы проводили. Тут можно сказать, что термин нокаут не совсем правильный, потому что поскольку в общем, структура достаточно сложная, и можно делать, вот, например, в случае, мы можем не обязательно нокаутировать, мы можем изменить, например, таргет-специфичность. И мы хотели посмотреть, в принципе, что мы можем получить. Например, для некоторых мы видим форму, что у нас есть форма делеции. Так, секунду. Вот, например, в данном случае мы имеем делетированную форму, которую мы видим, что созревает и наблюдается в клетке. И раз она у нее, как мы видим, здесь делеция как раз guide sequence области направления, узнавания РНК-мишени. И, возможно, у нее изменена мишень, и она теперь направляет метилирование в другую мишень. И...
Случайно, возможно, лучше не кажется. Мы уже 15 минут опаздываем, давайте оставим кулуары.
Да, хорошо.
Дискуссия, это же не вопросы, это...
Коллеги, да, действительно. Сейчас у нас ответ и постерная сессия. Я вас всех ждем на продолжение. Самое интересное впереди. Обязательно приходите в 15:20.